All right, guys, sorry I cut the last PowerPoint short. I forgot that we were doing 6.1 and 6.2 today. So here you go, here's 6.2. It's shorter than 6.1. I'm basically going to walk you through a biozone and sort of explain it to you, and then tomorrow you'll go through the tutorial from 6.1 and 6.2 and sort of tie it all together. So 6.2 is on DNA replication. We've talked about DNA replication before, so it should be familiar to you. The enduring understanding, again, is that heritable information provides for continuity of life. And in this unit section, the learning objective is to describe the mechanisms by which genetic information is copied for transmission between generations. So DNA replication is copying DNA onto another strand of DNA, okay? This happens, we talked about this when we talked about mitosis, because DNA replication has to happen before a cell can divide so that each new cell has an exact copy of DNA. The essential knowledge that we're going to talk about in this section is that DNA replication ensures continuity of hereditary information. So basically, all cells in our body have the same DNA. We're going to talk about the 5' to 3' direction when we do the biozone. We're going to talk about how replication is a semi-conservative process. That is, one strand of DNA serves as a template for a new strand of complementary DNA. So I'll show you in the next page what I mean by that. But if you imagine a double helix and you imagine unzipping the double helix, each strand of the double helix serves as a template, and two new strands are formed by the complementary base pairing rule. So then you get two identical copies of DNA. We call that semi-conservative. The third part here is an enzyme called helicase. Is an enzyme that you actually might need to know. Helicase is the enzyme that unwinds the DNA strand. So that's like the zipper. If you imagine unzipping it, that's done by a protein called helicase, an enzyme. Topoisomerase is an enzyme that relaxes the supercoiling in the form of the replication fork. So imagine you had a string, and you know how if you get a piece of yarn or something, the yarn is coiled up, but you could separate the pieces of the yarn. So if you were to pull those pieces of yarn apart, it would start to coil tightly in the area that wasn't unraveling. It would get a lot tighter. That's what happens with the DNA too. So topoisomerase is an enzyme that relaxes that supercoiling and allows the DNA to continue uncoiling. A third enzyme called DNA polymerase is an enzyme that synthesizes the new strands of DNA continuously on the leading strand and discontinuously on the lagging strand. We'll talk about leading and lagging when we do the biozone, but DNA polymerase sort of carries itself down the DNA strand and adds the complementary base pairs based on the template strand. And then ligase is the fourth and final enzyme that you need to worry about. And ligase is responsible for joining the fragments together on the lagging strand, which we'll talk about when I have a picture for you. So at this moment, if you'd like to pause, we're going to go over biozones 101 and 102 on the next two slides. So if you want to get a head start and do them on your own first, feel free. I recommend it, but I know you've got a lot of stuff to do today. So if you'd rather just let me walk you through it, that's fine as well. So pause here if you want to do it on your own. Otherwise, here we go. All right, so here is Biozone 101. I know it's kind of hard to see on this screen, so if you want to open it up in your Biozone book, pause and grab that and open it up. This is showing you the semi-conservative method of DNA replication, which produces two identical copies of DNA, each containing half the original material and half the new material. So if you look at the picture, you have the black and white double helix, which is open, and you can see the black strand and the white strand now have a blue coil with it. So you can see it's semi-conservative. Half of the new DNA is conserved and half of it is new. And on the other strand, half is conserved and half is new. So that's what we call the semi-conservative method. As we said, before a cell can divide, it must double the DNA. It does this by a process called DNA replication. This process ensures that each resulting cell receives a complete set of genes from the original cell. After the DNA has replicated, each chromosome is made up of 2 chromatids joined at the centromere. And again, this should all be review for you. The 2 chromatids will become separated during cell division to form 2 separate chromosomes, and during DNA replication, nucleotides are added at the replication fork. And you can see the replication fork at that little arrow that says helicase at the replication fork. It's basically where it's, if you picture this as a zipper, it's where the zipper is going to continue unwinding. And then it talks about the enzymes that are responsible for it. So step 1 is unwinding the DNA. What you need to know here is that the enzyme helicase is responsible for unwinding. Step 2 is making new strands. So if you look at the diagram, you see DNA polymerase on the left-hand fork, and it scans the strand and adds complementary base pairs on. And notice you've got at the top of this diagram a 3' and a 5'. That's going to become important. So DNA polymerase catalyzes the condensation reaction that joins adjacent nucleotides. What that means is it just, if it sees a T on the old strand, it places an A on the new strand. Sees a G on the old strand, it places a C on the new strand. The strand that's being synthesized, that's being made, the new strand, the blue strand, is synthesized in a 5' to 3' direction. So it adds base pairs in the 5' to 3' direction, which means that it's reading the template strand from the 3' to the 5' direction, and it adds base pairs as it goes. All right. Polymerase moves 3' to 5' along the strand that it's reading. Thus, nucleotides are assembled in a continuous fashion on one strand, but in short fragments on the other strand, because DNA polymerase can only go one way. It can only go from 3' to 5'. It can't go backwards. So if you take a look at this diagram here, and you look at the strand that's dark gray, notice at the part that's been unzipped, that's the 5' in the dark gray strand, and DNA polymerase can't hook onto that 5' end and travel toward the 3' end. It can't move that way. So what has to happen is, as this strand unwinds, helicase is unzipping it. DNA polymerase will attach here, and it will synthesize this strand. And then it'll attach further down, and it'll synthesize another little fragment. And then it will attach further down, and it will synthesize another fragment. So instead of this one that can hook right onto the 3' end and synthesize continuously the whole way down, this side, it's synthesis, and then it skips forward, synthesis, and then it skips forward, synthesis. So you get this broken chain for the new strand. That's what we're talking about here. When we say that they're assembled in a continuous fashion on one end and fragmented fashion on the other strand, these fragments over here on the lagging strand are then essentially glued together by another enzyme called ligase. And then step 3, rewinding the DNA molecule. Each of the 2 new double helix DNA molecules has one strand of the original DNA and one strand that's newly synthesized in the blue. And the 2 DNA molecules rewind into their double helix shape, and now you have 2 new chromosomes in a semi-conservative fashion. All right, so if we go to the questions here. Question 1, what's the purpose of DNA replication? Essentially, DNA replication prepares a chromosome for cell division by producing 2 chromatids that are identical copies of the genetic information for the chromosome. And then it does that with all the chromosomes, and they can split, and each new cell will have the same DNA. The main steps, you can just read the previous page and summarize those in your own words. Number 3, for a cell with 22 chromosomes, state how many chromatids would exist following DNA replication. 44 chromatids, sister chromatids, and they'd be glued together in pairs. Number 4, state the percentage of DNA in each daughter cell that is new and the percentage that is original. 50% is new, 50% is original. Number 5, what does it mean when we say DNA replication is semi-conservative? We explained that a lot, but it basically just means that each new double strand has one strand that's the original parent DNA. Number 6, how are the new strands of DNA lengthened during replication? This is when DNA polymerase adds nucleotides in the 3' end of the new DNA strand, matching bases on the parent DNA with complementary bases on the new DNA strand. Number 7 asks what rule ensures that the 2 new DNA strands are identical to the original strand. And the base pairing rule that ensures the correct complementary nucleotides in the DNA are paired with the bases on the parent strand, producing an identical copy of the original DNA. This is also called Chargaff's rule, where A pairs with T and G pairs with C. Number 8, why does one strand of DNA need to be copied in short fragments? I think I just explained that earlier, but basically DNA polymerase can only run one direction. It can only run from the 3' to 5' end. So on one strand, it has to be done in short fragments. And then number 9, match the statement in the table to form the complete sentence. Then put the sentence in order to make a coherent paragraph about DNA replication and its role. So I'll just read you the paragraph that they have. Doesn't mean that if you did something different, it's not right. But they put in the answer key that DNA replication is the process by which the DNA molecule is copied to produce 2 identical DNA strands. Replication is tightly coiled by enzymes. The enzymes the enzymes also proofread the DNA during replication to correct any mistakes. DNA replication is required before mitosis can occur. After replication, the chromosome is made up of 2 chromatids. Each chromatid contains half original and half new DNA, and the chromatids separate during mitosis. All right, so if you have any questions with that, let me know. And then for page 102, this is showing you all the different enzymes that control DNA replication. The only ones that you need to be familiar with are the ones from the previous page that we talked about. So for number 1, describe the general role of enzymes in DNA replication. Here, enzymes catalyze the reactions that occur during DNA replication. They unwind the DNA, so that's helicase, copy the DNA strands, that's DNA polymerase, rejoin the DNA sequences, that's ligase, and proofread the DNA to correct mistakes. That is also a different DNA polymerase. There are several different DNA polymerases. Where am I here? All right, we'll get to that in a minute. Okay. Then it says enzymes are important in ensuring the same sequence of DNA in the parent cell occurs in the daughter cell so that the cell functions normally. And then for number 2, state the specific role of each of the following enzymes in DNA replication. Helicase is responsible for unwinding the parental strands. DNA polymerase 1 hydrolyzes the RNA primer and replaces it with DNA. So there's a primer that you can see right, I can't read it on your screen, but primer is sort of right around here. And it, hold on, give me 1 second. Sorry about that, guys. Okay. So anyway, there's a primer that basically just tells the DNA polymerase where to start adding base pairs. So DNA polymerase 1 hydrolyzes that primer and replaces it with DNA. DNA polymerase 3 adds nucleotides in the 5' to 3' direction. So elongates the leading strand and synthesizes new Okazaki fragments, which are on the lagging strand. Those are those short fragments that get put together. And ligase joins Okazaki fragments into continuous lengths of DNA. And then number 3, determine the time it would take for a bacterium to replicate its DNA. The answer here is 4 times 10 to the 6th divided by 4,000, or 16 minutes and 40 seconds. And number 4, how is the energy for incorporating the nucleotides into the strand provided? For this one, the nucleotides are present as deoxynucleoside triphosphates. Hydrolysis provides the energy for incorporating them into this strand. All right, so hopefully that gave you a little bit of a background in DNA replication that's a little bit more in depth than what we had done previously. At this point, follow along with your plans, and tonight your homework assignment is to complete page 100 in Biozone. Post a picture of it and your model. So post a picture of Biozone and your model. And then on Friday, your homework is to begin your guide for the, what did we call them? The tutorial. So you're going to go to Unit 6 in the portal, and you're going to look at the DNA structure and replication tutorial guide on the right-hand side. You'll download that. You can either answer the questions right on there on your iPad, or you can print it and write the answers in. Then you're going to follow this link, which is also linked on my website. And as you go through, it's going to give you a checklist. There are going to be a lot of questions to answer, things to summarize, things to read. If it says read, just read it and put the check mark in. If it tells you to summarize, obviously do that. You might be filling in the blanks. You'll also come across quizzes. You'll have to sign in using the sign-in. If you click the upper right-hand corner on the quiz, you can sign in using the sign-in that that I sent out on the portal. And that's the only way that I will get your scores. So you have to sign in to take the quizzes. If you can't sign in, send me a message. You have to sign in to take the quizzes. And this right here says your answer is submitted as soon as you type it. You cannot delete it and retype. Be aware of this and be careful. That is true. However, if you get something wrong, it's not just automatically wrong. If you get something wrong, it's going to be given to you again. So make sure you understand the answer, remember the answer, so that when you get the question again, you can answer it correctly. So these, even though the quizzes are annoying, there are some that are pretty long. They'll give you a good grade in the grade book. So let that give you, hold on, one second. I'm almost done right here. That will give you a good boost of confidence there, hopefully. So any questions, let me know. Hope that was helpful. And this is the longest you'll hear me talk going forward. So I guess that's good. All right. Have a good night, guys. Bye.